the talk that on herpes virus that occurred in the interim actually is a pretty good introduction to what I'm going to talk about here. I'll start with the final hypothesis, which is the following. Here we have viral DNA packaging, either phage or, as you know, herpes virus is a homolog. So this could be herpes virus DNA packaging also. And in our experiments, we have found the possibility of an expanded structure that has would have to have a thinned shell, and then you have the final virus. If this was a herpes virus, the hypothesis goes as following. This sheet structure, this structure here, this thin structure, would have to be a sheet structure of some kind. And we're proposing it's a structure called Alphasheet that most people don't know about, but which I'll describe soon in more detail. Being a sheet structure, it will be reactive with a lot of different types of proteins if they have the same sheet structure. And the hypothesis says that amyloid proteins convert to alpha sheet structure and then react with this intermediate to neutralize it and stop it from going further. And that results in innate immunity if the process is healthy. If it's not healthy and there's too much production of amyloid alpha sheet, then you start piling up toxic material as shown on the right here. That's the overall hypothesis. So let's now go into some of the details. First, Alphasheet. This is from a paper by Pauling and Corey. On the left is antiparallel beta sheet, and this is Alphasheet. Alphasheet is derived from beta sheet in 2 dimensions by simply rotating every other amino acid by 180 degrees, and the alpha sheet has all positively charged groups on one side of it and all negatively charged groups on the other. This is an old paper. It was written in 1951. And this is a Wikipedia version of Alphasheet colored. Okay, important is that Alphasheet in human proteins only occurs in very short runs. One of the reasons for that is it's thought to be toxic if greater than 6 amino acids. And where do you find Alphasheet? You find it in potassium channels, water channels also, and in ion binding sites. Now note that in the Alphasheet is bent in ion binding sites, and in general, the reason you don't know about Alphasheet is that there are steric clashes that tend to occur with the side chains, and those steric clashes are neutralized by bending. So that's an important take-home lesson from this slide. Alphasheets tend to be bent. Okay, now let's get to that expanded particle, hyperexpansion. Here's a potential pathway, and over here is an electron micrograph of particles that were isolated as follows. They were first purified by buoyant density centrifugation in cesium chloride, and then buoyant density centrifugation in a nicodens density gradient. They were generated by a T3, phage T3 mutant. And nicodens does not enter these particles, so they band at a very, very low density because they're basically bags of water. And because they're impermeable, they look like they're full, but actually they're empty. There's no DNA in these. There's no DNA in these particles. To give you an idea of how hyperexpanded they are, here's a normal size capsid right there. And some of them are actually some of the mutant capsids are actually contracted. Here's the SDS gel profile of the different density fractions of the density gradient. They're all the same as the normal capsid 2. So this was a little bit frustrating that we couldn't really see that the shell was thinner because of the impermeability. We eventually found a particle where you could see that. This is actually a phage T3 head. We don't know exactly why you can only see it with the head. And it's a little bit hard to see here because of the severe range of contrast. Here's a normal capsid, and here's a phage. And then these are mostly expanded particles. You can see the hyperexpansion by cryo-EM also. Here is expanded, and here are normal sized heads. These are heads, so they're actually full of DNA. Okay, so what's the take-home lesson here? The take-home lesson here is these hyperexpanded particles exist for T3, possibly also for herpesviruses. We are not certain about whether they're intermediates in packaging yet, but for the purposes of the discussion here, it really doesn't matter. Okay, so what happens when you look at what's going on inside of an infected cell in vivo? Now, these cells are infected inside of an agarose gel. They're actually plaques. We thin section plaques of phage T3. And if you stain with urinal acetate and lead, you see these hyperexpanded things here. One of the reasons there are so many of them is that the number of these things is increased in gel. The number of hyperexpanded capsids is increased by at least 10-fold relative to conditions in solution. We developed a staining procedure for these hyperexpanded gels that's pretty much specific for them. It's an acidic phosphotungstic acid stain. And here's a combination of urinal acetate and acidic phosphotungstate. And you can see the phosphotungstate is actually precipitating around a hyperexpanded particle, which it only has precipitate over part of it. So why would you increase the number of these hyperexpanded particles when you grow in gel? Well, we don't really know the answer to that, but we have some idea that it might have something to do with the fact that bacteria, when they grow in gel, whether that's an agarose gel, an agarose gel, all your top agar that you use for phage plating, the bacteria are growing in that gel only because they're pushing the fibers of the gel aside. Though the pores inside of an agar gel, any agar gel that anyone has ever used for phage plating, are too small for the bacteria. The bacteria grow in there by pushing aside. And here's a thin section actually showing E. coli pushing aside agarose fibers, but it would be the same for agar. Okay, so how does this all relate to herpes? Well, it's known that there is a correlation between the onset of Alzheimer's disease and previous infection with herpes virus. And that correlation exists only if the ApoE epsilon 4 allele is present in the person involved. And that ApoE epsilon 4 allele tends to decrease the amount of collagen 4 and I'm blocking out the name of the other protein, both of which are cross-linkers that strengthen gel. So herpes virus infection is correlated with Alzheimer's disease only if you have a weakened gel structure. And I would propose that weakened gel structure is causing the cells, including Schwann cells, where herpes virus does grow, to be pushing gel aside as it's being infected. So that could well be the reason for the correlation of the ApoE epsilon 4 allele with Alzheimer's disease. And by the way, NIH has on their website basically a bounty on deciding why this correlation exists. So I think they might not need a bounty. It might have been done for free. Okay, so what does this all imply for what goes on inside of a person with Alzheimer's disease, what goes on inside their cortex? You would predict that there's Alphasheet present, and you would have to correlate this with the fact that, yes, there's a lot of sheet structure in Alzheimer brain, but it's not Alphasheet, it's beta sheet. And this has been known for a long time. That's basically the way you diagnose Alzheimer's disease post-mortem. Okay, so if this hypothesis is correct, inside of cells, there should be both alpha sheet and beta sheet, the alpha sheet being the toxic form, and the beta sheet being a conversion product which is a result of the cells trying to detoxify the alpha sheet. You might find the alpha sheet inside of lysosomes also, another way to detoxify it. So we obtained some human postmortem cortex, been fixed, and looked at thin sections. And this is what we saw. There's dark matter. There are inclusions. Lots of inclusions. These are almost impossible to miss. Almost impossible to miss, I say, even though they have not been reported before. There are dark inclusions, and there are light inclusions that seem to be embedded in the dark inclusions, and even lighter inclusions within the light inclusions. The light inclusions are decorated. It's as though these were all one inclusion at one time and have subsequently differentiated. The dark inclusions are very fibrous in nature. This is a good illustration right here. You can see there's all kinds of fibers. The fibers are not particularly well aligned, and based on what I just said, you might suspect that they were based on Alphasheet, because Alphasheet tends to bend and it would not align very well. A lot of this dark inclusion material is inside of what appears to be a lysosome, and in this particular person, there were lots and lots of empty lysosomes. This lysosome appears as though it's dead. There's nothing else in it besides inclusions. There are also inclusions outside of the lysosome. It appears that the lysosomes are trying to inactivate this stuff and are failing. In one way in which it's apparently being inactivated is being converted from dark to light inclusion. Here's a mitochondrion, which will show you that this is intracellular, not extracellular. A very interesting aspect is that even though these are eccentrically and irregularly outlined, there tends to be a matching of them, and the next slide will show you that in more detail. Down here is a series of dark and light inclusions. Almost looks like a jigsaw puzzle. And it's very hard to imagine how that occurred unless this was all one structure at one time and the light and dark parts differentiated later. And that's the way a jigsaw puzzle is also made. And here's a higher resolution view of the dark inclusion showing the fibrous structure and a light inclusion with the sub-inclusions in it. Okay, so let's see, where does all this come from? Here are some key previous studies. This is one by Claudio Bandea. He proposed that Alzheimer's and other neurodegenerative diseases are basically innate immune responses. His argument basically was that the amyloid proteins are analogs to virus proteins, and this is basically a CRISPR-like phenomenon where the cell takes up its enemy and then uses it to neutralize the enemy.
Philip, you have last 3 minutes.
Okay, this is almost over. And here's the paper showing the correlation of Alzheimer's disease with previous herpes. And here's one showing that the age-normalized incidence of Alzheimer's disease has been decreasing for the last 25 years in Europe and US, but not in Japan and China, and not and is being decreasing more for women than for men. Healthcare was suggested, and it turns out that these are all the vaccines that have ever been developed. So if you look, what vaccine was developed in 25 years ago? Herpes virus vaccine. What do you think? Good chance that's the herpes virus vaccine that's causing the age-normalized decrease in Alzheimer's disease. Therapy by phage capsid, possible, but you have to have persistence first. And here's an experiment on persistence. As phage T3 and T4 are highly persistent, T7 and 201 phage 2-1 are low persistence phage. Okay, I had to speed through that last part. I'm finished. Hopefully there's time for a question or two.
Thank you for another brilliant talk, and really nice to see a kind of human health side of things. I don't know if we have any questions yet, but perhaps in the interest of time,